we can conceptualize evolution, molecular evolution, as a walk in a fitness landscape. Schematically, in XY dimensions of the fitness landscape, there are all genomic sequences or all sequences of single polymer. And on the z-axis, there is a fitness of a given sequence. So you may have peaks, like here, and in one peak you have sequences that are very similar, and supposedly also they have similar structure. Peaks could be connected, like here, and what it means is that you can evolve from one peak to another peak through single point mutations. If a peak is isolated, like disconnected with other peaks, like here, then this kind of evolution is impossible because there is no connection through functional molecules, and selection, Darwinian selection does not operate on non-functional sequences. So we assume that the single point mutation mechanism is one of the major mechanisms in evolution, and certainly was the major mechanism in early evolution. Another major mechanism in early evolution was through acquiring length. So as a polymer gets longer, it gets more functional, and this pushes evolution forward. And there are two models for that. One model is this accretion model, when you start with some structure and then build on the success of this structure by adding additional parts. Another possibility is that as a polymer grows, it restructures, refolds into a different structure. And both have very different implications, but they are both very important because accretion model is essentially one of the pillars of modern phylogeny. On the other hand, this refolding or restructuring model is essential to explain what we believe is true, but we don't know for sure, that molecules that a small number of initial structures evolved into this bewildering array of functional polymers. And otherwise, we would have to assume that each different structure had a separate evolutionary trajectory. So we have a number of questions, and they are very fundamental. Was evolution through single-point mutations likely or even possible? Surprisingly, there is really very little, if any, evidence that this is indeed the case, even though we strongly believe it, at least for early evolution. Molecular evolution. What is the significance of neutral or near-neutral mutations? These are the mutations that bring you from one peak on the landscape to another peak of the landscape. If peaks are very closely connected, it forms a network, and then full evolution is possible and evolutionary optimization. But if peaks are separate, then you know that evolution is largely governed by chance, because where you start determines where you're going to end up. And there are these models, theoretical models, kind of spherical horse models, that tells us that for sufficiently long genomes, everything should be connected through networks. But experiments that are pretty much the only experiments that have been done on mapping the landscape by Irene Chen showed separate peaks. So what's the story on this? And then what's the efficiency of improving function by length? Are any of those accretion or restructuring models true? So the experiment. It's a very kind of classical experiment. In some way, it's a classical experiment in molecular evolution. We take a piece of RNA and a substrate, and the reaction is a reaction in which the substrate, a low RNA fragment that acts as a substrate, is ligated or joined to this piece of RNA. So this large piece of RNA acts both as an enzyme and as a substrate. So it's a little bit of a weird reaction. It's a kind of single turnover reaction, but why is this so, you'll see very shortly. And we use RNA now, in contrast to my dear friend Antonio, we are quite unsure about the existence of RNA world, but no matter what, it's a wonderful tool to study evolution. And that's how we use it. So we actually looked at two sequences. One is 20 nucleotides, and the other one is 80 nucleotides to explore the influence of length. And for 20 nucleotides, we have 10 to the 12th possible sequences for 8, and we can synthesize all of them. For 80, of course, it's totally impossible. So we just sample things at random. So here is the experiment. It's a typical RNA evolution experiment. When you start with this fragment random sequence of 20 or 80 base pairs, there are some promoter and constant region that are necessary for studying for the experiment to go, but that doesn't play a role in what I want to tell you. So forget all about it. Then you do transcription, and you create this large pool of RNA molecules, and 10 to the 16th altogether, and incubate it, mix it with a substrate. And most of them will do nothing, will not react with the substrate. Some of them will react in the substrate, and because they react in the substrate, they will be longer, because the substrate is attached to the enzyme. And that allows us to very easily separate those that reacted from those that did not react. That's why the experiment was constructed the way it was. So those that reacted, we can amplify, make more copies, and go through the rounds of this. Amplification could be accurate or could be done with mutations. We can regulate the level of mutations, and with mutations it would diversify our pool. And you go a number of times in this cycle, and then you look what you got. And here is what you got. So first of all, you see that even for a very short polymer, 20 nucleotides only, we do have catalytic activity. So short polymers are capable to be catalysts. For 80, we have even more catalysis, and these are what you really see here is this is the sequence. These days we can sequence like millions of sequences, and we do. And here is the one that was discovered the most often. That is sort of expected to be proportional or related to their catalytic efficiency. It's not as simple as that, but it's some measure of catalytic activity. So there's a second, third sequence, and so on. And some of them are in green, some of them are in other colors, and both you have some in green. And why they are both in green, you'll see in a moment. This is not just random choice of colors. So if you have, let's say, look at this sequence, it kind of looks like, okay, there are two stems, junction, at the junction, and a little loop for 20 amino. Black is for the 20 nucleotides kind of enzyme. For 80 nucleotides, you have something very similar. It's just that instead of this small loop, there is a bulge and a stem with a loop. So this motif got a little bit bigger, but there is no question that it's essentially the same motif that got bigger. So it is consistent with this accretion model, something that people were looking for and had never found before. And you see a very similar thing, like another sequence of 20, which is very similar to this one, and another 80 that is very similar to this one. If you look further, there are some longer sequences that I gave you for reference. You see that, again, it's a very similar motif. And even this is kind of the same motif but kind of richly decorated, but, in science, like with art, decorating something too richly doesn't make a better result. So this is not really as efficient as this one. So we see, just to summarize this part, we see that we have that we have actually first example of an accretion model when we can show that when we go from 20 to 80, it just is the same motif but slightly improved. In fact, to follow on this, we just investigated lengths between 20 and 80 and found out that that this motif is conserved and the fitness improves as we would expect, at least between the best selected polymers of different length. So now let's talk a little bit about there are some other sequences. This was just an example of one sequence. So let's of one type of sequence, one peak in this landscape. So let's talk about how the landscape looks like in general. So we can cluster the sequences. Now let's be clear. The way we cluster sequences is based entirely on the sequence, on single point mutations, which means that in a single cluster, there are sequences. If you take any two sequences in a single cluster, you can get from one to another through intermediates that are all active and differ by single point mutations. So, but it doesn't say necessarily that everything that is in a single cluster has the same structure. So, and here is an example of that is indeed not necessarily the case. This is for 20N, and there are two different sequences that differ by 11 steps, single point steps, but they're connected in space. And if we take, let's say, A as a reference point and do some measure of how similar are everything in this cluster is to this sequence in terms of structure, what the exact way of measuring similarity in sequence in structure is not really relevant here, but what you see is that there are two peaks. So and if you start from the opposite direction, we also see two peaks, and they are kind of connected. You see that they're connected, but they're peaks, so there are two different structures within the same cluster. So evolution from one structure to another is possible here, because they are connected. And if we go to ATN, the situation is quite similar. Here we just have four different clusters that account for about 85% of all structures that we sequence, and there were millions of them sequenced. And so we have this four clusters, but if you look at those clusters and do this measure of secondary similarity in secondary structure, you see that they are not necessarily the same structurally. So there are two peaks, three peaks, and so there are several structures that are connected. However, between clusters, situation is not nearly as simple, because we have clusters differ quite a bit. So 36 to 44 mutations to bring you from one cluster to another, and we don't have a real path between clusters. But within cluster, again, you see that there is this structure A and structure B. These are sequences similar to A, and these are sequences similar to B. So again, we see that there is refolding restructuring is possible. So let's conclude what we learned from this exercise. So first of all, even very short polymers can act as catalysts. 20 base pairs is quite short for a function that is not necessarily that simple, like ligase. Some short and long ligases share functional motifs. So that confirms this accretion model, and to my knowledge, this is the first evidence for this accretion model. Now, we find these near-neutral networks and we find that they were much more extensive than in the experiments of Irene Chen, but we did not find this extensive networks that were postulated by in theoretical models that everything is connected with everything else. Now, it doesn't mean that it isn't. Remember that for these 80 nucleotides, we could only probe a part of space, and different experiments will have to be designed and will be to check whether it's possible to connect different clusters. And protein refolding through single point mutations is possible, which tells us that evolution through single point mutations is possible. You can move from one point in the fitness landscape to another point in fitness landscape that is not necessarily very close through single point mutations, but also that we see this refolding or restructuring which is the essence of this restructuring model. Again, that had not been proven before. Thank you.